The following is a conversation with Dr. Lydia Mapstone, a microbiologist by training, currently working as co-founder and CEO of BoobyBiome. BoobyBiome is a startup biotechnology company that's working in the field of the microbiome. It aims to produce new live biotherapeutic products for preterm infants. This is Inside Matters. My name is Dr. James McElroy. I hope you enjoy it. Dr. Lydia. You're a doctor now, right?
I am a doctor, yeah.
Yeah. When did you get the PhD?
When did I get it? Well, I did my Viva in April last year.
Have you just like erased it from your brain?
I honestly,
You're like, oh,
no,
trauma.
No, I love my PhD. I loved it. It was just really stressful having a company at the same time, as I'm sure you can imagine. Just trying to balance them both.
I understand.
And there's so much failure in both companies and PhDs.
Right.
So when you combine them together, it's even more interesting.
And you're going to tell me the story now. I thought you'd pressed pause on the company for a while. No,
it was
it's even more impressive.
Background.
It's even more impressive.
Ticking, yeah.
Right. Well, before we get onto that great story, I'll take you back a step.
Okay.
How did you get interested in what you did your PhD in?
Oh, my PhD. I'm a biologist, so I did my undergrad degree in biological sciences, which is really general. So it covers, I think I studied mangroves, bats, ants, a bit of human, not much though, plants, just very, very broad. And then I found microbes the most interesting in my undergrad degree. Mm-hmm. I think actually when I first really learned about how important microbes are in those communities was when I had a tutor. When I was a little first year student, I had a tutor and she was really interested in bats, and she got us to write essays on bats. And one day she's like, do you know what, guys? Instead of having to write an essay on a bat this week, I've decided to open it up and you can do an essay on caves, anything you want about caves. And I think she was hoping we were all going to, talk about bats because they're the most interesting thing in caves. Right. So she thought. And then I went
but it's the bugs in the cave.
It's the bugs in the cave.
Yes, of course it is.
It's microbes. How are they doing that? How do they survive? So I did a whole essay on all the different microbes that live in caves. And do you know what? I don't think she even read it.
All right.
I didn't get marks back.
That stimulated
me and made me I just found it really interesting. And they went and did a master's in synthetic biology, biotechnology. And I just think synthetic biology and microbiome is so cool. We're not a synthetic biology company at BoobyBiome, but we're very interested in that. That's really my background, SynBio. And just microbes, what we can do to microbes to get them to combat the different challenges we've got. So in my master's project, I got involved in something called iGEM, which is the world's largest synthetic biology competition.
Oh.
And we sort of made a team, and it's a bit like having a little company. So you have a team, you've got 3 months to build something in the lab, and then you go on and you pitch it at the competition, and then you go win prizes, and there's lots of different people get involved. And my team got really excited with bacteriophages. So not quite microbes, but bacteriophages. And we come up with this.
So for the listener, these are tiny viruses that infect bacteria, essentially.
Yeah, yeah.
And they're inside of us.
Yeah, there's loads of them. Way more of them than bacteria.
Right? Yeah.
Oh, yeah. Yeah.
So what did you do with phage then at iGEM?
We thought it'd be really cool because bacteriophages are very specific to the bacterium. So a bit like how, cold viruses only really infect us. It's the same with bacteria. Each bacteria strains even can have a particular phage for them. And so we thought it'd be really neat if we could engineer bacteriophages to deliver a CRISPR package, which is a sort of like molecular scissors, which can snip up bits of DNA. So we engineer the phages to deliver, they infect the bacteria, and then the bacteria then have this sort of delivery for sort of enzymes inside them now, which will then cut up antibiotic resistant genes on their plasmids.
Oh, nice.
So trying to stop.
So you weren't trying to use phage to kill drugs which were resistant to antimicrobials. What you were trying to do was remove the antimicrobial gene cassettes from existence.
Yes.
So just remove all it.
Yes.
Because it's...
And that protects the microbiome a lot more as well in that respect.
Interesting. So for the listener then, bugs become antibiotic or resistant to antibiotics because they somehow acquire or develop a toolkit which allows them to produce substances which block the antibiotics from having their action on the bugs, right?
Yeah. So a lot of bacteria, not all of them, but a lot of bacteria have their genomes where, that's where they make all their proteins from and how they live and survive. And but then they also can have circular pieces of DNA called plasmids, which can be quite short. They're just small circles, and they will contain often huge different number of genes for breaking down different antibiotics if they're exposed to them. So most of their genes for antibiotic resistance aren't on their genomes. They're on these little bits of circular DNA, which they like to swap and shoot out at each other and swap.
Right.
And swap with each other. And that's how you get, huge amounts of antibiotic resistance, because you get, different bacteria swapping all their favorite genes. And then they end up having a whole cassette of these plasmids, these circular bits of DNA of all of the different genes for every single antibiotic in them.
One of the challenges with antibiotics as well is that because the body is such a reservoir of diversity, if you like, you might destroy lots of bugs that you want to destroy.
Yeah.
But there might be a tiny number who have those cassettes, those circular pieces of code, and they survive.
Yeah.
But because they survive, they replicate, and then suddenly you've got a larger critical mass, right?
Yeah, exactly.
Yeah. Fascinating.
Yeah.
But the phages would resolve the issue of AMR potentially.
Yeah. Potentially. That's what we were trying. Well, that's what we sort of showed. We only need 3 months to do it, but it was really fun. We won the best therapeutic prize as well, which was great.
Boom.
That was it was my first sort of hint of how you could use biotech to try and, do something good. So actually, as well as that first phage that came in that cut up the antibiotic resistant genes, we then also had a second bacteriophage to mop up the ones which didn't get targeted by the first one. Trying to beat evolution that way. So that was quite a complicated project. I don't really know.
For 3 months?
Yeah. But
did you do any scientific work?
There's a big team. Yeah.
You did some scientific
There
work?
were 9 of us.
It wasn't just a concept, actually?
No, we did it in the lab. We built these. We engineered these phages.
Goodness,
it's amazing how much you can achieve in 3 months. But we were a team of, there are 9 of us, all working full time. And we were lucky enough because we were at the University of Edinburgh at the time, and that's got lots of really great PIs and academics that could, come in, chip in, give us a piece of advice. Give us a phage.
Amazing.
Let's work on it.
Why has no one? Maybe they have.
They are. Ligo bioscience.
Oh, Xavier.
Yes. So I recently went to a webinar with him, and he was describing, and I was like, he was describing what his main, innovation of his company is, and I was, so I did that. I did that as an iGEM student ages ago. Yeah. I mean, obviously he's taken it. He's actually making it work.
Oh, absolutely. Yeah. It's weird. I messaged him this morning.
Really? That is weird.
Yeah, just to say how you're getting on, because I met with him in San Francisco in January. When you were saying that, I was thinking that sounds an awful lot like illegal.
Yeah.
But maybe it was different because I'm not an expert at all in the gene editing or phage. I just know that phage are really important.
They're very cool. Yeah.
And could be the future.
But
the phage companies, like, and Xavier won't mind me saying this because he positions himself as a sort of synthetic biology company rather than the microbiome company per se. They've not really done it. The phage companies. There isn't a standout, as far as I'm aware, knock it out the park phage company anywhere. I think there's really big manufacturing challenges with phages. I mean, how do you grow the phages?
So what you do is you'd get your bacteria that the phage like to infect. You grow it up, and when it's sort of really happily doubling really frequently, you add your phage to it. And then what happens is
they chew them all up and then you just extract your phage.
Yeah. So they just go.
But what happens to the bacteria that they sort of shoot themselves into? That's like clunk and debris that you need to move away, isn't it?
That's true. Yeah. So they lyse the.
Yeah.
There's 2 types of phage. So the one that we were having that was engineering the first bacteria, this was called a lysogenic phage. So that actually wouldn't cause the cells to burst. That just sort of sits there. A bit like those little bits of DNA, those plasmids it makes.
So it sits there and they just sort of like shrivel up and die, or what happens?
Well, the DNA gets passed on to the daughters. It's a bit like how a plasmid was made, probably. It probably came from a phage.
Oh, interesting.
They just pass it on.
And they stay static then? So they're like bacteriostatic phages?
Yeah, they'll just stay inside the bacteria. And now and again, they might mutate and become a lytic phage.
So they're just there for the ride?
Yeah, they're just there for the ride. Yeah.
That's crazy.
So that's how it works. So the first one would.
That's crazy. So there's a microbiome in the bug. That's not an unreasonable analogy. I've talked about this before. There's a picobiome.
Yes.
Or is it a nanobiome?
Ooh.
What's smaller, nano or pico?
Pico.
Pico's the smallest, so it's a nanobiome then. Yeah. So the nanobiome are all the phages that are kicking around, just chilling in the cell cytoplasm, in the cytoplasm of the bacteria.
Yeah.
Okay, you heard it here first.
But I mean, you can't get too many of them because if you get a bacteria with too many of them, they're using all their resources, aren't they? They're using all the cellular machinery to replicate and
But surely they just replicate to the point where they burst the cell, or
Yeah.
is there some sort of control within?
Yeah.
Okay, we're getting really technical and phages now,
Yeah,
which is
we
great
are.
because I'm not.
Well, I'm not an expert either. Ages ago now.
You're an expert.
Blind leading the blind. But yeah, no, it was just an exciting project. And I really enjoyed it. And it was a really good way for us to experience for me to realize you could make something and it potentially could have a massive impact. And then.
And did it trigger some entrepreneurial flair inside of you?
I would say so. Yeah, it was fun.
Particularly because you won, right? You were like, wow.
We went the overall winners. That's a very prestigious prize, but we did win the best therapeutic prize, which was important.
That's a great accolade.
Yes. And then why I picked my PhD was because I then did a PhD. I basically wanted to live in London. That was purely not particularly because I'm clever reasons, but I was just like, you know what, I really want to live in London. So
PhD looked good. And
I just bought the project. Well, I got into a really great program called LIDO, which is the worst acronym ever, but it's a PhD program that is run and it's all across London. There are different universities which offer projects, and then you pick from a portfolio of about, 200 different projects, which one you want to do. And so that's why I just read 200 different projects, and I just thought I want to do something in synthetic biology and microbiology still.
Yeah.
And that's why I picked my PhD topic, which was nothing to do with the microbiome. Well, sort of, same techniques, but not really microbiome. It was to work out for us, to come up with a system for genetically modifying and also seeing if we can cultivate a species of algae. Mm-hmm. Well, technically it's called a cyanobacterium. It's a blue-green algae called spirulina, so that we can make a space crop for astronauts when we go to Mars.
This is so the thing
in a nutshell.
That I hope we do get to Mars, but so when people get to Mars, they're going to need to eat.
Yes,
and
exactly.
They're limited by basically what they carry over. And one day there might be shuttles just going back and forth all the time. But this would be some sort of fresh food.
Yes, well, I mean...
Is that as good as it's going to get on Mars?
What, spirulina?
Genetically engineered spirulina.
So those that haven't
Earth suddenly sounded really
before, good. it can be best described as swampy. So yeah, this and sulfurous.
I won't tell you what you said about the juice.
No.
I'm hoping we'll sponsor this podcast. I won't say no more because, No.
But yeah.
Swampy is good, by the way. Swampy is a compliment.
Yeah.
For in my opinion, for someone that's wanting to...
for a good gut.
Yeah.
Yes. No, I completely agree. with that. And the spirulina is amazing. And I'm just quite sad that spirulina isn't one of the ingredients, actually.
Also swampy. You've not actually drank from a swamp.
No, well...
Maybe you have. Okay, right. Fine. So spirulina is swampy and you have to take it in a capsule.
But it's really, really good for you. It's got all of your essential amino acids in it, for example. So you don't have to eat different proteins. You can just eat spirulina and it's got balance of all the essential amino acids. It's also got the same amount of protein.
Hold on a second, because I think you've listened to Andy Scott on the podcast, the bodybuilder that was so big you couldn't fit through the door. Also my personal trainer when I'm in Glasgow. And you can imagine how fun that is. He's of the view that you just can't get big like him if you don't eat meat all the time, and there's no vegan bodybuilders who are as big as the ones who have animal-based protein because it's more complete. But it sounds like it's not more complete. It sounds like spirulina has everything you need.
Yeah, although to get the quantity that you need of protein, I mean, it is really high protein content spirulina. So if you look at, for example, insects, if you were to eat insects, because that's another big thing for Mars as a potential food source.
Insects and spirulina. Wow.
I know, they've got a lot to look forward to with astronauts. Spirulina has the same amount of protein as insects if you on a gram for gram basis.
Wow.
But why that's incredible is because insects are a higher trophic level. So insects have to eat something like spirulina or grass and then gather up their protein. But spirulina, cutting that, and it's way more energy efficient because you get the same amount of protein.
Yeah.
But
you don't have to digest all the fascinating.
And it's got all the essential minerals.
So it's superfood.
Yeah, it is a superfood, and it does deserve that name. But there are some massive limitations to spirulina. It's still quite hard to grow. It's hard to grow it in a dense enough culture.
So when you have spirulina powder, that must be derived from a culture. It's an algae. So somebody needs to create like a big vat or like a big system.
Pipes are quite good because you need a big surface area so light can penetrate.
And they just scrape off the spirulina?
Oh, you probably filter it. It's quite long. It's quite a sort of...
Oh, is it?
Sort of matte. It sort of floats. Yeah, and actually, it naturally occurs in alkaline lakes. So it's sort of... in Chad, for example, as an example, and it sort of floats to the top and forms these big slimy mats, and then people just gather it in and then eat it. And, and it's really good. They found that people, the locals there that eat spirulina, they're
very healthy.
30% less likely to get vitamin A deficiency, even if they're locals and they don't eat it.
Really?
So yeah, and it can make up a huge amount of their diet as well.
Wow.
But,
so what did you manage to develop a method to grow it efficiently over your PhD?
So, well, I was trying to see if we could grow it with the resources locally available on Mars for astronauts. That was one part of my project.
Did Elon Musk call you and say...
Do you know what? No, I should have really reached out to him in hindsight. That was a mistake. Yeah, maybe.
That's his vision.
Yeah.
Maybe he needs some spirulina in his life.
Yeah. That's probably one of my biggest regrets, not sending him an email. It would have taken 5 minutes.
Hey, Elon,
why didn't I
we got something for you. But surely the outputs are still there. You could go back to it if you want.
Yeah.
You're busy now with your venture, which we're going to get on to. So what was the output then of all of that? And how did you decide that? Because not many people have done what you've done and what you're doing, which is decide to start a company during your PhD in a subject that doesn't actually link to your primary topic of research, right? That's the case. It's different. So how did that happen?
Well, I think I realized from iGen, but I really wanted to get involved with a startup, or
Why was it the buzz?
Yeah, it was so fun, and
buzz.
I just felt we could move really quickly, actually, even though we were a small team.
Yep.
And it just seems like we can make a big impact. And I think from that, I was like, well, do you know what? I need to learn what it might be like to be in a startup.
And you like the feeling of winning as well, presumably, right? Yeah.
That's fun. Although, to be honest, I've learned how to fail a lot.
Yeah,
because we don't win a lot. We mostly don't win.
I mean,
we mostly lose.
I think my first prize was £500 at the University of Edinburgh pitching competition.
Oh yeah.
And I sort of ran back, just like, yes, get it. I was like, that's so good. I'm going to be able to do so much with that.
Was that with your company, or was that with a different?
This was the very, very early days of enterobiotics, like the earliest of earliest days. Like it started in a 24-hour computer laboratory in the University of Edinburgh. I was trying to find a topic to write my dissertation on.
Yeah.
I was not having much joy. I was doing a very nerdy form of procrastination, which I was going from medical journal to medical journal, landed on a Nature paper that showed that if you move bacteria from a thin mouse into an overweight mouse, the overweight mouse loses weight.
Yeah.
I was like, whoa.
Yeah.
That's honestly what happened. You just made the noise and that.
Yeah.
I mean, there's not much going on in my brain anyways, but it just sort of, something happened. Yeah. And I was like, that is unbelievable. Because I tried to start a personal training business beforehand, which failed miserably. One client, and that was my mum. And so I stood up and I just, I won. And I just had this idea. I was like, I wanted to do this fecal transplantation thing, and I won the prize. And I was just so excited. I was like, I've got to do this now. So it's amazing what something like that can do to your confidence.
No, completely. Yeah, so actually really similar to how BoobyBiome started, really similar. So I sort of was like, well, do you know what? It'd be cool to try and see if we could get a startup or get involved. And so I just came across a competition that we could do as PhD students called Biotech Yes, which is just have you heard of Biotech Yes? Oh, okay.
Maybe.
Well, it's mostly for PhD.
Where is it?
Not really,
Where's it based? France?
It's not in Nottingham. No, it's just a UK-wide competition. Where they try and encourage PhD students, postdocs to come up with a mock company, and then it's a 3-day workshop where you meet experts, and then you do a pitch competition at the end. And then if you go through that, you go to the finals. And then
sounds fun.
Yeah, it's really fun.
You meet lots of interesting people.
Yeah. So I mean,
but how many people from Biotech Yes are still going with their company?
Not that many.
Probably very, very few.
Not that many. Yeah. But the ones that are, they're doing well.
That's good.
But yeah, they but I was like, okay, well, I want to do this competition. Looked around me, let's try and get a team together. So I got a team together, and then we were going to the pub over that summer before the competition. And we're like, right, what area?
That was a testing ground.
Yeah, it was actually. Yeah, it was a good testing ground.
Good testing ground, yeah.
We were sort of like, what area can we, can we innovate?
Someone said boobs.
Actually, no. Well, yes. Someone did say boobs, but that was later. That was me just sort of looking about the different areas, and there's so many different things going on in modern life. But as a microbiologist, microbiome's always been really interesting. And I just came across a 4-page document called Post Notes, which the government releases every year. They're 4-page documents on areas which the UK government thinks we should innovate in science and technology. And one of them in 2018 was on the microbiome. And there was just this one line. So I was reading about the microbiome and I was saying, we need to really innovate in the microbiome. Here's the reasons why microbiome's interesting.
Oh.
And it's a really interesting document, looking back at it now. But there was just one line in that note where it just said, the breast milk seems to have microbes in it, which is interesting. And babies not breastfed are more likely to get, autoimmune diseases. And I was just like, what?
Yep.
That was
a brain explosion.
Yeah, because I just assumed it was sterile. I didn't realize that there was a microbiome in breast milk.
Right.
I mean, now there seems to be a microbiome
basically nothing sterile.
Yeah, exactly.
There's a microbiome in everything, including blood.
Yes, yeah.
Which is another mind-blowing thing. Yeah. But anyway, sorry, yes.
Yeah, so I've read that.
That blew your mind.
That blew my mind. And we're like, well, what is in this breast milk microbiome? And there have been a number of papers published, but it wasn't included in the Human Microbiome Project. And the level of detail.
And for the listener, that was a big deal, wasn't it? That
Yes.
was funded by the National Institute for Health in the US, focused on trying to map the microbiomes of the body, essentially, right?
Yeah.
And it didn't include breast.
No.
Wow. Okay.
So the level of detail just wasn't that high of exactly what bacteria are living in this breast milk microbiome. How do they get there? How does it differ? There wasn't a lot of detail at that point in 2018. And so we just thought, well, you know what, this is classic women's health in particular being overlooked, underfunded. Let's see if we can innovate in this space. And so we just came with that idea. We weren't really planning on taking it forward, but we just got such good feedback from that competition. We just sort of looked around, and actually we were a team of 5, and we were all doing PhD students. We were all doing PhDs, and me and my 2 co-founders that took it forward were the ones that sort of were like, we stayed up really late the night before the pitch. We realized we worked really well together.
Okay.
And the other 2 amazing scientists now, both really good academics now.
Yeah.
But they decided, for them it wasn't.
They wanted a different
them.
life.
But that was fine. We're still really good friends with them. And like I say, they're very successful academics in their own right. But yeah, we took it forward with 3 of us, and we just started doing competitions.
How was the pitching competition though, by the way?
Oh, the first one?
Yeah.
Oh, awful. We lost. We didn't even get through to the next round.
Why? What happened?
I don't know. No, there was just another company that was better than us. Also, interestingly, but they didn't go forward with it though. They didn't win either, which is sad.
That's sad.
But yeah, they also, the one we lost to, were doing acne treatment using microbiome.
Okay, topical.
Which is very relevant, to be fair.
Very, very relevant.
And I don't think they went forward with it.
I mean, acne is driven by the microbiome.
Yeah.
It's just bugs that are getting caught in the glands. I can't remember what they're called, sebaceous glands or something like that. Yeah. Fascinating.
So we didn't win, but that was our first taste of failure, but also first taste of actually excitement.
And at that point, were you already in your head fine at the idea of, or whatever, actually doing this alongside your PhD? If something was to come of it, you were going to do it at the same time, because that's a huge thing.
It's one of those things you do a little bit. Like, oh, this is an idea. Let's just see how far we push it.
Yep.
Just push it a little bit.
Yep.
We'll do another competition. Why not? We didn't win.
What was the original pitch idea? Can you remember?
Yeah. The original idea, and maybe we will do this one day, was to sample women's breast milk, look at the exact microbial composition in her milk, and then if they're missing out on any key species, we'd supplement to give a very personalized product.
So if you now supplement
with the bacteria back from our culture collection,
and it would find its way into their breast milk.
Oh, sorry. Then we should mix it with her milk and then feed her infant.
That makes a lot of sense. Okay, that makes a lot of sense. Got it.
That was our original.
So I was like, how does it get there? But how does it get there originally?
Oh God. Everyone asks this question, and no one's entirely sure.
No one knows.
There's two theories.
Okay.
Theory number 1 is that it just comes from the infant mouth. So when the baby's feeding, it just sort of, the bacteria in the baby's mouth goes back into the ducts and it crawls up. And there's been a bit of evidence for this, if sampled baby's mouths.
But I suppose, just to push back a little bit, could it not be that it's the other way?
Yeah. So that's theory number 1.
Yeah.
But theory number 2.
Interesting.
Is a thing called bacteria translocation, and that's when bacteria hitch a ride on an immune system cell and sort of travel through the lymph system to the ducts. And there's been there's been evidence both for this. There's been evidence where they made these poor, sad, germ-free mice. These are mice which haven't been exposed to any microbes, and they fed them a probiotic, and then they got them to have baby mice, and they found the bacterium that they fed that mouse showed up in the mouse's milk. So there's quite a lot of evidence of it.
It's like a sci-fi thing.
It is.
They hitch a ride on an immune cell and then go, yep, this is where I need to be.
Yeah.
And that might be some sort of incredibly conserved evolutionary thing that just happens with all humans, which again is another one of these, my mind.
There's just so much we don't know though. But, there's so much more research needs to be done on this. And it's also, it's one of those things where, if it's really difficult to research it. And, our techniques for culturing and assessing the microbiome are still pretty new. So sometimes people ask me, do you think if a mother was a C-section baby herself, does that mean that she's passing on, she wasn't exposed to the first vaginal microbes of her mother? Does that mean that her infant microbiome won't be as good or will be less diverse? And you've just got to be like, we haven't been able to study it.
Right.
So it's really exciting. As we start to do these longitudinal studies, we'll get more answers on that. But right now, these techniques weren't really around.
So you're talking about the analytical toolkit that we have.
Yeah, absolutely. So,
and it has come on a long way though, hasn't it?
Oh, yeah.
We were, up until relatively recently, trying to grow everything on plates.
Mm-hmm.
That's all we had.
Yeah.
And microscopes.
Yeah.
But now we've got next generation sequencing technologies.
Yeah, it's changed everything.
PCR, So, okay, cool. So it started to snowball. And it's funny, as you're telling the story, I'm thinking, God, it's the same as me. It was a super exciting thing.
Yeah.
And I think if you'd shown me a crystal ball and said, yep, in 2023, you'll be employing almost 70 people, you have an MHRA license, you'll be just about a phase 2 study, I'd be like, what? Surely not. Like that's, I'd be like, that's mad. Yeah. That's insane. Yeah. And it sounds like that's what's happened with you. It's just like, it's trickled and then it's just become this huge thing.
Yeah. Yeah.
You're going to get to the point probably in the story where it became almost unmanageable to do the two.
Yeah, it was pretty unpleasant at times. Well, it was amazing. So we managed to get a very little amount of money. So we managed to get 50K to work on our first sort of proof of concept. We started collecting milk. And with our PhDs, we were really lucky actually. So me and my two other co-founders. So I'm
Can I ask you, sorry, just I'm sorry to interrupt you, but you were talking about the original idea.
Yes.
And then we were going to get onto what it became. And that's probably what you raised the 50K for, is that right?
Yes.
Okay, so what was then the idea that it became that you got some money?
Well, it was when we realized that the logistics of trying to do a personalized medicine approach for microbiome was just going to be an absolute nightmare.
One day.
One day.
So cool, right?
Very cool.
Yeah. One day that would be the thing to do.
And we started looking more into the different babies which are most vulnerable to having dysbiosis, so an imbalance in those beneficial bacteria in the gut. And we realized there's three different sort of camps of babies. There's obviously the babies that aren't given any breast milk. So actually 40% of the first bacteria in an infant's gut come from breast milk. So it's a huge input of these first beneficial bacteria. So of course you'd think, okay, formula-fed babies, obviously they're missing out on breast milk. So they're missing out on these first really important
colonizers.
Early colonizers, yeah. It's been estimated it's about 30% from your birth delivery as well. So it's 30% from when you're born, from if you're born naturally, you're born, from your mother's vagina, and you're coated in all those microbes. And then of course, if you're lucky, maybe she'll have a little poo while she's in labor if you're lucky. So you might get some of them as well. So it's one of some of those bacteria. And then the next seeding is from how you're fed. Wow. And then there's going to be about 30%, which is just.
But just think about
a dog.
If you have
skin,
two people or two babies, one's born through C-section and bottle fed, the other's born through normal vaginal delivery or just vaginal delivery, whether it's forceps or not, and is breastfed.
Yeah. There's
a 70%.
Yeah.
All else being equal.
Yeah.
70%. Yeah. That's huge.
Yeah. And they are much more likely to have different diseases like asthma, obesity is one, diabetes. But interestingly, they can also be absolutely fine. And that's what's so
as is always
about microbiome. Because you can't, their genetics or maybe their dog was really good in the house that they had. And it's really something that we're spending a lot of time doing.
Dogs are a thing though.
Yeah.
I know it is.
Yeah. Well,
dogs influence your microbiome for sure, right?
Yeah, I mean, I did a parasite course in my undergrad about dogs and cats, and I don't know, it's really put me off. But you're right. They found that babies that are exposed to dogs growing up have more diverse microbiomes in early childhood.
Yeah. Incredible.
But yeah, sorry. So where was I going with this?
Really interesting. So you were talking about how the original idea wasn't viable because it was too difficult to scale.
Yeah.
Because logistically, you'd have to get a sample from every single mother, figure out what it's missing, using a variety of tools, and then also replace what's missing, which might be different from a mother to mother basis, and that's a different product for every person.
Yeah.
Okay, so that's just
yeah,
not feasible at the moment.
Yeah. So that's when we started looking at, okay, well, let's if we want to refine this a bit more, what are the different babies that actually are at most risk? And it's if you're not given any breast milk because you're missing out on that those beneficial bacteria, formative ed. The next camp is if you're born C-section, which we've touched on now. So C-section babies, they're missing out on those first sort of vaginal and GI tract microbes. And then the other group baby, which are the most vulnerable, arguably, are preterm babies. So if you're born too early, it's really difficult, firstly, for mothers to produce enough breast milk because the milk's not ready to come in.
Yeah. Physiology is not sort of developed enough yet because of where they're at in the whole process of having a baby.
Also, these babies are often on very high levels of antibiotics, as is the mum as well.
Right.
And as a result of that, they have very, very poor microbiomes. And to be honest, their stomachs also aren't, their guts aren't developed properly. So often they've got a leaky barrier, and the tight junctions in their guts aren't close together. So things can sort of slip through to their bloodstream. So they're very vulnerable group. And
how important do you think tight junctions are and leaky gut is?
Well, why are you asking about evil glints in your eye, James?
No, I mean, I have my own view. I don't want to. I don't want to influence your thinking.
No.
There's no right or wrong answer to this, by the way. But
so, I mean,
evil glint. I'm not plotting or scheming anything. I promise. Yeah.
So I think with these preterm infants, it's fairly important because it's just undeveloped. So the crypts. I'll just smash the mic then. The crypts aren't anywhere near as developed as well. They're sort of stubby. You're meant to have lots of nice long. You probably know what the term is. I always forget it.
They're called
crypts of
crypt abscess. No,
in your gut, high surface area.
I know exactly what you're talking about.
Yeah. But they're just not developed very well, and it means if you do get a nasty opportunistic bacteria pathogen coming in, it can just very easily just slip through those gaps and go into the blood. So I think for preterm infants, it is pretty important.
I think I totally agree with you, and I don't know why I was making an evil blend. I think they're important for everybody.
Yeah.
I mean, for adults, arguably just as important or more important. I mean, when that barrier starts to break down, then they do translocate and influence local and systemic inflammation, and that kind of leaky gut, quote unquote, has been linked to all manner of problems, both in the gut and beyond the gut. So, figuring out how we can sort of get them tighter again through microbiome modulation is a key area of interest for me, for sure.
Yeah.
And I hadn't appreciated that it was a big thing in preterm babies, but it makes total sense. And it's probably the fact that they're preterm, and probably the fact that they're getting loads of antibiotics.
Yeah.
Right? And it's just creating an entirely not normal situation or environment for them.
Yeah, absolutely.
So, and there's a lot of C-sections and a lot of preterm babies, right?
Exactly. Yeah, it makes it even harder for them. So, I mean, there's a few diseases because of this, but preterm infants are particularly vulnerable to.
Yeah.
One of them being necrotizing enterocolitis, or NEC, which is a very unpleasant disease. So actually, I should say as well, about 10% of births are preterm, and then roughly about 10% of preterm infants will develop NEC at some point. And this is a really nasty disease. So like I say, it's when an opportunistic pathogen comes in, enters the bowel, and the immune system, which hasn't really been trained how to respond appropriately, responds with really high inflammation, and then you get necrosis of the lining of the gut.
Death.
So death. Yeah,
that's a bad thing.
Very bad thing, because then you get leakage of the gut into the abdomen, and it can have really high mortality rates.
It can be fatal.
Yeah.
And probiotics have been used.
Yes. So there's been loads of trials where they've shown that probiotics can reduce the incidence of NEC happening. And there's been no other treatment for it, really, to date. It's not used in hospitals. And the best thing, if you've got a preterm infant, to try and reduce this is to give them breast milk. And in hospitals, if you're really lucky, if you've got a preterm infant and you can't breastfeed, your baby will be given donated breast milk, which is next best thing. However, it's been shown that actually your baby's still not quite as well off as if it was to get your own milk. And I think a big part of that reason is because it's been sterilized, the donated breast milk. So you are not receiving.
So it's a bank of breast milk.
Yes, exactly. So they're receiving donated breast milk that's been pasteurized, which makes sense because you don't want to give any. There's no chance of the infants getting a undesirable microbe coming in. But
so they get the nutrients, the calories, the micro and macronutrients.
Yeah. Some of the immune system elements survive, not all though.
So there's some immunoglobulin in there.
Yeah, but it's a bit varied, but none of the bacteria.
Yeah.
Yeah.
And that's what we're talking about, it being so important.
Yes, exactly.
So does that mean that there are big programs, like volunteer programs, where women donate their breast milk?
Yes, there are. Yes, yeah. There's one in London, it's the Heart's Milk Bank, which is a really good one. But yeah, it's really helpful if mothers can overexpress, if they do overexpress milk, then they should try and donate it if they can, because it's the next best thing at the moment.
It's really interesting.
For preterm infants, yeah.
And I presume, it's only for preterm infants. It's not something that people would buy commercially. No.
None of that.
It's too scarce a resource.
Yeah. I mean, you can't, there's some weird dodgy stuff on Facebook, I think, where you can buy breast milk, and bodybuilders love that. Apparently some do in America.
My goodness, that's ridiculous.
I know, and I think they can drink. Yeah, that's not good.
Right, well, next time we get a bodybuilder on here, I will ask. Yeah. They probably won't readily disclose that they've been looking at buying that to get.
There's been these weird Netflix exposed documentaries on the breast milk, weird breast milk trade. On sort of,
really?
Yeah.
So there's a whole underground black market for breast milk.
Yeah, it was just yeah. It's not good.
Wow.
Yeah.
Okay.
It's a bit unfortunate for us because we're trying to sampling very widely, but we can't. We're not going to use that. Obviously, we need to be there at the sample site and make sure it's done properly.
100%.
And the consent forms and
100%. I mean, particularly if that's so. You're going to get on now to what you're doing at BoobyBiome.
Yeah.
And at what point in the journey did the name kind of form?
Right at the beginning, BoobyBiome was sort of like a joke name, to be honest, but we love it. It describes what we do, and it's if we want to keep parents on board and mothers, I mean, they all love it. If you do market research on people that probably are going to want to buy our product, it's parents, and they think it's great.
Right.
Not everyone loves it. It's a bit of a Marmite me, so admittedly, BoobyBiome. It took my dad quite a long time to say. I kept on calling us BoobyBiome.
Oh, really?
No, Dad, it's BoobyBiome. BoobyBiome.
Is he there now? Is he kind of on board with it? Okay.
Yeah. He says it now.
That's good. So BoobyBiome is doing supplementation for preterm infants, or is
so we're really right at the moment we're gunning for trying to create a product from the breast milk microbiome that we can supplement to preterm infants in hospitals, with the hope that we can then reduce the incidence of neck in particular, but just their health in general. If they can put on weight faster, and just avoid other diseases such as late onset sepsis is a big one as well for preterm infants. That's really what we're trying to do. Make a life.
Must be so scary for parents, late onset sepsis.
Oh yeah.
Oh my goodness, that must be frightening.
But we are still researching, and right now we're just very wide. We're running around London collecting breast milk. Unfortunately, it's mostly had to be London just because our labs in London, and this milk needs to be really fresh. Because as soon as we get milk, we take it back to the lab. We try and culture directly from it, and then we also extract all the DNA and look at what community structures there. But we want to do that quickly. We want to keep as many of these bacteria. And we've got a really big culture collection now for different, very interesting microbes that we've isolated from breast milk. And from that, we're sort of designing our synthetic community.
Are they very... So is the idea that you'll have a consortia?
Yes, absolutely. Yeah.
And that consortia will be sold as a product, a drug or a probiotic, depending on where you go with it.
Yeah.
Right?
Yeah.
Are you on the fence about that still, or you can...
It is a difficult one. I think it's a question that most microbiome companies have, because it's such a new area of the microbiome. The regulation behind it is difficult, because historically these probiotic products they're seen as food supplements. They still are seen as food supplements. Having said that, in the EU, no one actually has been given the status of a probiotic. So you can't say you're a probiotic in Europe if you're selling a live bacterial product. It's not been regulated. In America, you can, but there's a definition of what a probiotic is, and it says that you may have to make a positive impact on health. And to date, even though I think there's been over 200 different claims to the food safety authorities in Europe and the UK to try and get that status, that claim, and no one's managed it yet. No one's managed it. No, it hasn't been granted for anyone.
So I think you're talking about the WHO definition.
Yes, sorry, yes.
Like, I think the WHO one, which is like, probiotic is a bacterium that, when administered in sufficient quantities, confers a health benefit to the recipient, or something like that.
Yeah.
Broadly speaking. And I think what you're saying is how many of the probiotics.
Well, this is what's so frustrating because,
Right.
you know, there's these probiotics that have just been around for decades and they're really good at making cheese, for example.
Yeah.
And they're not really doing, you can take them, but they're not going to do a lot. But then there's some probiotics that have been really intelligently designed. So I'm going to name drop. Simproof, for example, is an adult probiotic you can take, it's full strains. And it's in liquid formulation because that's the best way to do it. And keep them in liquid, keep them alive. And then, and you drink it. And there's been loads of clinical trials with them, but they still haven't been able to get this probiotic health status. And they do this huge amount of research that they've done to show that it's having a really great effect on your health. So it's very frustrating, but you can see how tempting it is as well as a product, you know, as a company developing bacterial product to go down this easier, less stringently regulated route of being a food supplement.
I was speaking to someone at the conference that we were at earlier this week, which is a microbiome conference for the listener, and someone was saying that they describe the difference between live biotherapeutic products and probiotics as basically probiotics is more about your marketing spend and how much you can market and sell and get influencers and so on. Whereas the LBPs is more about convincing regulators and ultimately payers, whether it's the NHS or some sort of insurer in the US, for example, rather than the patient themselves who would be buying a probiotic off the shelf. That's what they were saying. But also, if you think about it, LBPs are probiotics by the definition.
Essentially,
so it's quite confusing.
Yes, it is very confusing. So, I mean, in terms of the preterm infants in hospitals, I think you got to get this right. You've got to do all the research. You have to do clinical trials. So really, that's the route that's what we're aiming for.
You're going to do a clinical trial in a preterm infant population because that's going to be difficult. Right?
Oh yes, it is going to be difficult, but it's not. There is a precedent for it. You know, these preterm infants have been given probiotics in the past, and actually, a lot of them do look quite promising.
Yeah,
but you know, they can reduce neck by 50%. I think we can reduce it by a lot more with a more intelligently designed product.
Well, in episode 1, we had Dr. Richard Hansen, who's a pediatric, he's a pediatrician.
Yeah.
And I think has an interest in neonatology. So like basically babies that have just been born.
Yeah.
And what he said, I recall, was that actually probiotics were really good for neck.
Yeah.
But still, for some reason, it wasn't being used
widely. No, not at all.
In neonatal units around the country, which just it seems a bit weird. Maybe if it was a drug and they wrote a script for a drug, perhaps people would be providing it more, because I think there's always a bit of resistance from the medical profession. Also, there's a lot of snake oil.
Oh yeah, completely agree.
I'm not going to say who it is, but I've been going back, and maybe you've seen it on LinkedIn, I've been going back and forth with this one probiotic company, just been like, can you stop? You're saying your product is backed by 300 or whatever it is studies.
Yeah.
No, it's not. What you're doing is taking one of the strains that's in your probiotic mixture and doing a PubMed search and then deciding that as a result of everything that's been published by other people.
Yeah.
Not in the context of your consortia.
Yeah.
Do you know what I'm saying?
That's so frustrating. Yeah. I mean,
that makes it difficult.
We go in Spooks, we buy probiotics on Holland and Barrett, and we culture them in the lab just because it's just really interesting for us. And we've often get bacteria which actually, they're not even listed on the packet.
Really?
You know, it's like, oh, that's weird we picked up that one. Let's double check the packet, and it's different.
Generally speaking, my view, or what I heard, was that the bigger companies actually, if you buy one of them and it says it's got E. coli Nissel or something like that in it, it's probably got E. coli Nissel in it because they're big and they've got the quality control and so on. But there's still a big hesitancy, I think, within the medical community for probiotics.
Yeah, absolutely.
And that's because, as you say, originally it was, this is really easy to culture and it's stable. So this is a great candidate. Whereas what you're talking about is the future. And that is, let's rationally, intelligently design a mixture of bugs which have been designed intelligently based on their phenotype and what they're going to be doing within the recipient, essentially, within the microbiome. So it sounds like what you're doing is you're trying to understand as best as possible what the healthy microbiome in breast milk looks like.
Yes. So we're sampling a lot of different women, and I have to say it is variable, which is a shame. Some women we call them our super donors, have very rich breast milk with, probably similar to you.
It is, yeah.
You know,
yes.
they've got very rich breast milk with lots of different of these bacteria, which already in literature have been shown to be doing quite a lot of different things. You know, boosting the first immune system, for example, is a really important role of these first microbes that you get exposed to. And then some women, it's basically a desert. There's hardly anything.
Really?
There's hardly anything.
Have you correlated that to anything? Like, have you got any suspicions?
Yes. We're still piecing things together. So all our donors give them quite a long questionnaire, which we keep on adding to and being like, oh no.
Yeah.
But it's hard. We want to ask them the world.
What you're
we
doing.
want to spend them an hour doing it, but these are, you know, mothers that just had a baby.
Yeah.
So it's quite hard to try and get the most efficient questions out of them. But
what you're doing is really important. That characterization you're doing on both the donated milk, but also of the profile of the person donating is actually really valuable. It sounds like no one's actually done that before.
Oh, well, that's kind of used to say. Yeah.
Really important.
We've struggled to try and make all these different links. I mean, there's often a paper that's out that says, oh, if they were given antibiotics in this particular time period, then that means that their milk is less good. But then it's less good. That's, you know, less diverse with microbes. But for all these different other factors, what was their occupation? You know, so actually one of our super donors was a primary school teacher, which I think is really telling.
That's
probably she's been exposed to so many, you know, so many of these little cute children, and they're all germy and sneezing.
Germ factories.
Yeah.
The stool or gut microbiome in the super donor, have you got any idea if that correlates? Like, is there a correlation between gut microbiome diversity and breast milk microbiome diversity?
Do you know what? Just because of sadly, we haven't been able to take their stools. Very sadly. Maybe we should couple up and set up a project on your FMCs. Purely because when we first started, we just didn't have enough cash. And there's so many questions. So we get the baby stool sample because that's very important to see that these bacteria that we isolate from breast milk are actually showing up in the baby's stool. But we haven't been able to. Yes. Yeah. Oh, yeah. But we also, not all of them though. There's a lot, to be honest, this is the thing about microbiome as well. There's a lot of bugs that we culture, and we're just like,
what is that? That's
not doing anything, is it? Realistically, that's just on for a ride.
It's passenger.
It's going to die as soon as it goes into the gut.
In terms of like scale, I mean, like in the gut, there are probably like 300, 400, 500 different species.
Oh yeah, it's much less
typically.
In breast milk microbiome.
Yeah, it's like orders of magnitude less. Is it like, you know,
well, actually,
5 to 10?
About 200 species have been identified from breast milk microbiome, but it does vary. I mean, we don't see in some of us we might just see 5 different species, but then in others, our super donors, we tend to see a lot more. We tend to see between sort of 20 and 50. It doesn't really go much.
So the super donors are an order of magnitude higher in some cases. That's amazing.
Yeah, sometimes.
There's got to be something there.
Yeah.
And are the babies of the super donors more healthy?
So that's what we're trying to track. So that's why we track a few of them over time. It's
going to be a long study.
Yeah, that's the thing.
Yeah.
But yeah, I mean, right now we are sampling mostly from full-term mothers, instead of preterm mothers. But we
how do you find the mothers? Like, I mean, that probably sounds like a ridiculous question. There's mothers everywhere, but how do you identify?
Mostly from word of mouth.
Really?
Yeah, and we use apps. Facebook's amazing. There's also breastfeeding support groups. And next door, we decided not to go down the hospital route purely because it's, well, first of all, you're getting them all from the same hospital, which sort of limits your sampling quite a lot. But then, although you can get way more, but it also just the ethical consideration, it just takes a lot longer. There's sort of bureaucracy over having to get that all approved and through. So, and it's been pretty easy for us actually getting donors and donations. I think, you know, once you explain what we're doing, the research, why it's important to mothers, they're on board and they've got all their mates who've just given birth.
Yep.
That they also want to donate. So for us, it's been fairly straightforward. How do you find your donors?
Yeah, so very interesting. During the early days, it was very like, I don't know if first generation advertising is like the right phraseology, but it was like newspapers, email distribution lists, posters, and an element of word of mouth. As the program grew, and we became better at like social media, it moved to a more digital form of advertising. But we still found that the traditional forms of advertising were very high yield. Email being pretty good, particularly if you can get the right distribution list. So like everyone who works within a building who are quite health conscious, just as an example. And then we built the number 2 brand
Mm.
and the website.
Yes.
And we have vans now that drive around Aberdeen with number 2 all over them. And there's just been this kind of, not exponential because that's the wrong word, but as the program has grown and our reach has improved, it's triggered a cascade of word of mouth and sharing and WhatsApp groups as well.
Yeah.
You know, I went to the gym recently and started speaking to someone, and I'm now top to bottom in Inside Matters clothing wherever I go, or Enterobiotics clothing wherever I go.
Yeah.
So if it's hiking, I've got Arc'teryx stuff as well. Just because like I know if I speak to somebody, I can convert them to being a believer, you know, 'cause, you will believe in the microbiome. And interestingly, I went to gym and I was started chatting to someone, and he was a nutritionist and a bodybuilder. And he started to say, oh yeah. Just off the bat, one of my friends just started to donate for the stool collection program in Aberdeen. I was like, oh, what's that called? Is it number 2? I was like, number 2. I was like, okay, that's interesting. So how's it going? He was like, yeah, it's great. I'm thinking about doing it myself. Eventually I said, look, I'm running that company. But I was so excited to hear that someone was excited about donating their stool to the number 2 program.
That's basically like that moment that people get when they finally launch a product and when they go into the shop and they pick up their project, their product. You've had that now.
Kind of, yeah. I was really excited. I mean, this can go one of 2 ways. Yeah. Last weekend, and the person won't mind me saying this, I was in the Western Baths, which is where I go for the sauna and the plunge.
Okay, lovely.
And I was speaking to this person beside me in the sauna, and said just about to start his phase 2 clinical trial, and he said he was working as something, and he said, "Are you James McElroy?" And I was like, oh God. Like, yes. He was like, oh yeah, I applied for a job 2 weeks ago. No one got back to me. And I was just like, oh no. I was like, I'm really sorry. I then I ran out very dramatically and got my phone and brought it back into sauna, which you're not supposed to do, and I said, please add me on LinkedIn. I'll make sure we reply to you. The thing is, for some jobs, we get hundreds of applicants. And we don't have the resource because we're a small company to actually enable people to get good and proper feedback. Yeah. But it's the whole number 2 thing was really empowering for me, and I was really excited about it. I believe in the power of word of mouth still. We're not on TikTok.
Oh, do you know what? We're starting to go on TikTok.
Are you? Yeah.
TikTok, breastfeeding TikTok is massive. And that's what we find out so many things though from our doing it. It's like doing a massive sort of questionnaire. I mean, there's so many videos of mothers, for example, crying, so they've had to pour away all their milk because it's been left out too long and things like that. And you learn a lot about, you know, what the different culture is of.
I know gut health has gone like
it's huge.
Like it's gone bananas on TikTok. Yeah. So I feel like I should be on there, but I'm also a little bit scared about. I've never used it, but I, like everyone, I mean, I find LinkedIn addictive.
Yeah.
No, that's LinkedIn. It's like a networking website. Never mind TikTok, which is like a design. It's been designed to be addictive, hasn't it? Yeah. They've literally designed it to. Anyways, sorry. So going back to your donors, we're word of mouth and other things. We've just built an app for donors to try and make the experience even better.
That's cool. Yeah. I think I don't know if it'd be worth it for us just because we're slowly, because we've got right now a big collection mode, but then over time, of course, we get the same bacteria coming up again, and we're almost, you know. So
you think you're hitting a bit of a steady state now, and you're seeing the same thing over and over?
Yeah, potentially. Yeah, we're definitely, our massive discovery phase is we're mostly over that hump, but now we still get new ones now and again. But I think, for us, we've almost finished that sort of big collection push.
And a businessy type question then, I'm interested in why you decided you wanted to do it yourself rather than trying to find a group who maybe had existing or going to a bank, for example.
Oh, that's a really good question. I suppose there was no decision not to do that. That probably would have been a sensible thing to do. I think when we started it, it was just, let's just see, we've got an idea, let's learn some skills on the side and let this grow. But then, you know, we got the investment, and then we were like, oh, okay. And then we just happened to know a lot of mothers who just about to give birth or had just given birth. So we suddenly had actually quite a big pool already at our not disposal. Yeah. That sounds awful, but you know, ready to use.
Yeah.
And then we just got really excited, and we're a team of 3 founders. So it's me, so I'm the microbiologist, but then I've got my co-founder Shaunad, she's a chemist, but she did her background in cell biology and her PhD in cell biology and DNA origami. And then my other co-founder is Tara, and she's a neuroscientist, but loves bioinformatics. So we sort of had the skill set between the 3 of us, even though our PhDs went directly in the microbiome.
Yep.
We had a really, really great skill set for tackling the microbiome.
Yep.
And we knew exactly what experiments we wanted to do, and and we had a bit of time to do them actually, because during our PhDs we were allowed to take 3 months off to work full time to do to do that.
That's what was in my head then about not doing them side by side. But you still
so I had 3 months off my PhD to work on that, and then obviously PhD is 4 years long.
Yeah.
But we managed to sort of back them up. So it was me and then Shaunad and then Tara, and then we had enough data, and then it was just.
So you staggered the 3 months.
Yes, exactly.
Proper co-founding team.
Yes. They're amazing. Yeah. I couldn't have done it without them. So that's again balancing PhD with a company. I think I couldn't have done it without co-founders at all. I don't know how you managed it.
Well, I didn't do a PhD.
I think medicine degree, James. But yeah,
a lot of people say a lot of people with co-founders say, I don't know how you did it without a co-founder, but it's one of these things where when you're in the thick of it, you're in the thick of it.
Yeah.
And you just, if you want something badly enough, like you've got that unstoppable burning desire where you go to bed every night and as your head hits the pillow, you're thinking about it, and you wake up and the first thing you think about is, how am I going to do this?
Yeah.
You find a way.
Yeah. No, you're
that
right.
said, burnout's a real thing.
Yeah.
It is.
No, you're right. Well, actually, James, interestingly, you're going to love this.
Okay.
Being very nice to you now. I went to a talk that you gave at the BIA Pulse program, which was in Francis Crick Institute years ago now.
When was this?
I think it was in 2019. Or no, 2019 March.
I just turned 30 and I've started to lose my brain cells. I won't actually remember doing
But you gave a really good talk, and you said, thing is, you have to give yourself time, because otherwise you're going to burn out. You have to let yourself go to the gym. And
okay, I'm still saying the same thing now.
It was great. No, but it was really empowering, because you get so many stories of, co-founders and people, my thing's about to fall off, talking about how they get up at 5 a.m. and then they work, and then they, head hits the pillow, and then they sleep 4 hours and they do it again. And it's just so, I just
not sustainable. And I don't believe people when they say they do that. So I'm really pleased that had an impact.
Yeah, it had a massive impact, because I think, to be honest, there were at points where we were doing that, but I think only not for very long.
You got to keep it going.
Yeah, you got to keep a routine of.
You got to have that green juice.
Yeah. Well, apparently, I can't wait to have the green juice.
You need to have some of that press green juice, which is really good. But that's really interesting, because that was now 4 years ago, and I haven't had a burnout in the last 4 years. I think I've been close to being at my limit. And sometimes I think I've gone too far in the gym training direction, like doing 2 or 3 Monroes in a weekend and going straight back to training on a Monday, and then doing the sauna on Monday evening, and then doing 2 gym sessions on the Tuesday, one with Andy Scott, the bodybuilder. And it's like, do I really need to do that? Probably not. But it's true. You need to think about, so in some respects, startup is a sprint. I'm sure you've heard the phrase, startups like jumping off a cliff and figuring out how to build the plane, and building the plane before you hit the ground, and taking off in the plane before you perish. It's kind of like that, right?
Yeah.
So it's a sprint in that regard. You've also got competitors, and now you've raised some money, and there's a burn. So you're burning, you're not burning the money, but you're spending the money.
You've got a big fireplace.
And one day that money is going to run out, and you need to have gone from A to B and maybe even to C before the money runs out, so you can raise more money at higher valuation. So in some respects, it's a sprint, but in other respects, you're going to be doing it for a long time.
Yeah.
And you need to make sure you're putting the work in most days. Otherwise, it's never going to happen, right?
Yeah.
So sounds like you guys have got that built into your...
Yes. Well, yeah. I mean, we work really well together as a team, and we know if someone looks at getting a bit burnt out, we're like, right.
Holiday. Yoga.
Yeah, exactly.
What are your burnout tips now that you're 4 years on from the BIA Pulse thing?
Exercise. So I run. I love running. I quite like running actually without music.
Interesting.
Which I don't know, it's more meditative.
Allows you to decompress your
you just don't even think at all. It's just like white noise in your head. So I also love cold water swimming. I know that sounds really like.
Oh, I love cold water.
I say, oh, do you? Oh, good. Okay.
2 weeks ago, I was cracking the ice and locking.
Oh, I love that. I love that. Yeah. So I swim. London's actually really good for cold water swimming.
I heard. Yeah.
There's lots of different spots. But yeah, I think like cold water swimming is so fun. I don't really like swimming in the summer actually. I just find it's not. You don't get the thrill out of it. Yeah.
It's a bit colder in Scotland. I'm not.
Oh no, I'm sure it is. Yeah.
Well, you're coming to this event, aren't you? In March, are you not the One Health thingamajig, the conference?
Yes.
Well, I'll give you some world swimming spots if you want when you're in Scotland. There's many locks that are cold. You're wearing a wetsuit or not?
No, it's cheating.
It's cheating. But
no, you know what? I wear the gloves sometimes.
Yeah, the gloves and I wear the gloves and boots as well. Yeah. Cool. Well, I think cold water swimming is fantastic.
It's just such a
So you do the Wim Hof?
No. I do it. Do you?
Yeah. Go. Breathe in. And out. And you do that 30 times, and then you hold your breath, and then you hold your breath until you can't hold your breath anymore. Then you take what he calls a recovery breath in, and then you squeeze the breath. And I've actually slowed my heart rate and actually stopped my heart doing this.
So cool. Maybe I should do that. I'm like, more just sort of jump in and try not to scream. And then...
No, that's what you do. You don't do Wim Hof when you're in the water.
Right.
You might pass.
Oh, okay. Yeah, okay. You do it before going in.
Yeah, before you go in, or just when you wake up in the morning.
Oh, okay.
Yeah, and it feels weird. But yeah, I know it sounds ridiculous. Like, why would you hyperventilate? Why would you do that? But like,
why do you do it?
Why would you intend? Well, I'm not going to say I'm an expert in the science still, because I'm not at all. So, but I think it's something to do with alkalizing.
Right.
Your blood.
Okay.
And triggering cellular spring cleaning processes.
Yeah, I'm trying to remember from Goop Lab when they sort of discussed this.
And also, so the high CO2 when you're holding your breath acidifies your blood, and that has some sort of benefit as well. Okay. I mean, Wim Hof is not just some crazy guy. I mean, he's mental. He is absolutely mental. Yeah. But he's also like a scientific physiological phenomenon. I mean, like they have done every possible experiment, and he just defies what people think is possible every single time. And that must be to do with him controlling his autonomic nervous system through his breathing and his cold water and everything like that. So I'm a big fan. In fact, I'm probably going to do a Wim Hof thing with Wim Hof.
Really?
It was one of my 30th birthday things. I was at this year. I'm going to do one of Wim Hof's sort of 2 or 3 day courses where you like do his method for a day and then you go climb some mountain in shorts.
Cool.
Which actually sounds right up my street anyways. So I don't know why I've waited so long.
But you turned 34 years ago.
I turned 30 last week.
Oh, what? Yes. I thought you said you turned 34 years ago.
I'm 30 years old.
Oh, wow. Well, happy birthday.
Thank you. January 24th, 1993. Yeah. Do I look, how can I look older than
No, you don't.
With all the stuff I do.
You said you turned 34 years ago.
All this pressed juice.
So you were 26 4 years ago.
I was 26.
And I heard you give your talk.
Yeah, 26.
Okay.
Yeah. I first started to get interested in antibiotics when I was like 24.
Okay, cool. So age to me, BoobyBiome.
Yeah, started antibiotics at 24. Yeah, it's been that long now. Oh my goodness. Yeah. I've got some grey hairs as well, but it's not too bad. Yeah. I've seen people worse off at 30. Sorry, we're now talking about my hair. And that's really off piece. We need to go back. We need to go back to BoobyBiome and what's happening next. So you've got a good collection now?
Yes, so we've got a big collection. We're testing all our different bacteria on, we've sort of simulated a gut in the lab and we're putting our bacteria on the gut.
How have you simulated a gut in the lab? What is it?
Well, with this one, it's not there's more and more sort of, what's the word? There's better ways you can simulate guts that get more and more complicated. We're doing quite a simplistic one where we're just growing the cells of a guts that line your guts in a petri dish, essentially.
Yep. And
just keeping them alive. But then, even these cells that line our gut, they can release immune system factors. They can, they can respond to inflammation. The barriers, the tight barrier junctions can sort of fall apart. It seems it's very simplistic, but it's sort of a very good, very early model you can do to simulate the gut in a petri dish. And we're doing lots of different experiments on our strains, seeing if they can reduce inflammation, for example, if they can make those gut barrier functions, get a bit closer and improve those tight junctions.
Yeah.
We're doing a lot. We've got matrix of different experiments we're doing and scoring our strains essentially, but we're also looking at competition assays. So for example, if you've got a nasty, because preterm gut, for example, has already often got these not great bacteria there present. And so we're seeing
why is that? Why are the bad ones there?
Yeah,
We don't know.
I think especially with all my C-section, the first microbes that get exposed to it often on surgical instruments, a hospital environment, which is just very unfortunate. But preterm gut is full of quite unusual microbes. They're not what you normally see in full term. But we're seeing if we can outcompete those. So if we put our bacteria onto our little simulated guts, can they?
Yep.
Can the other ones not be there? So we're doing a huge number of different experiments, and then from that we're sort of making a smaller and smaller list of actually what are the functionally relevant bacteria, which definitely are doing something, as well as can they survive into the gut. So formulation is a really important thing in microbiomes. So it's all very well, you can have your perfect mix of bacteria, and hypothetically they should survive to the gut because obviously we've seen them in breast milk and we've seen them in stool samples, but bacteria in the body can behave very differently from, bacteria that have grown up, been freeze-dried, been mucked about, and kept us got not great temperatures. So can they survive that and also then, colonize and live in the infant gut? So a huge number of tests. And then from that, we're going to go...
You're doing all of this in-house?
We're doing most of it. We're outsourcing a little bit. We've outsourced most of our... So as well as sort of testing these strains, of course, when we get breast milk, we want to see exactly what's in that community. And we're looking at very high level of resolution, what particular substrain is doing that particular thing in a breast milk microbiome. And we've outsourced that. Microbiome actually is supported by some really great companies that can offer really good services, but it's really come along.
Absolutely.
Yeah, I mean, we use Cosmos ID, who are great. But they can just really quickly, we just send them our DNA and they read it and they tell us what's in there. And that's really, really helpful. And that makes us so much faster and more agile. Otherwise, we'd have to have a very expensive sequencing machine in a house. And so we're testing all of our strains at the moment, and we're kind of working out what would be optimal. But as we're doing this, there's more and more papers coming out all the time talking about C-section babies and formula-fed babies. I mean, there's one...
That supports your narrative.
Yes, that supports our narratives, but also make us think about other products that we could potentially develop outside of preterm infants. So that is something that we're also keeping an eye on. I don't know if you've seen, there was a paper that was released in the end of last year where they found that C-section babies respond less well to vaccines than full-term babies. So it's really your first microbes that are having this really profound influence.
So even at that very early stage.
So even those C-section babies, their microbiome can actually... So actually at first it's very different, but then it will converge and look very, very similar to a naturally delivered baby's microbiome, especially if they're breastfed. But even over a similar age of one, that's that difference actually can already... It's already happened. It's your first bugs. It's your first training of the immune system. So that's very interesting.
This microbiome immune system link is the real deal, isn't it? There's
also with the research coming out formula for babies. So there's a paper published in February now, in January, where they found that babies that were given antibiotics, if they were breastfed, they're 3 times less likely to develop asthma than if they were formula fed.
3 times?
Yeah.
Wow.
So I mean, so we're keeping an eye on also what's going on outside of preterm infants, but we're really at the moment sort of trying to focus, because that's obviously very important, to try and not be a complete jack of all trades.
I presume that it's not possible to take a breast milk sample and expand it somehow, in some master bank.
Yeah, so that is an interesting idea. We have thought about that. And it could be possible. I think the difficulty is because there is still quite a lot of variation between donors. We'd have to be, and you don't breastfeed for that long often. I mean, the World Health Organization recommends that you breastfeed for 1.5 years, which is ages. I mean, when you first have a baby, and the first 6 months you're meant to be exclusively breastfeeding, and that's at least 6 times a day plus a night feed. So it's really difficult. It can be really painful. It can be really painful for women as well. So actually, our breastfeeding rates are quite low, but it's quite rare for mothers. I think the UK is particularly difficult because there's not much support, unfortunately. But after 2 months,
not much support for mothers.
Yeah, for mothers to continue breastfeeding, there's not much. It's quite difficult for some women to pick it up, for some mothers to pick it up and to continue. And it can be very painful. You can get infections.
Yeah.
And I think actually in the UK, it's about 50% by 2 months have stopped breastfeeding. So trying to get reliable donors is just slightly more of a challenge. And then even if it's really the first 3 months of breast milk that we're interested in, so that window's even smaller. And then even from that, it's just you'll have to screen it. I guess I know you guys do that for FMT, but it's babies, so we have to. The screening process got to be really careful with what's in there. So it is something we've considered, and maybe that might be a nice product down the line.
Because I'm just thinking, are there other benefits to the natural milk beyond
Oh,
the
of
bugs?
course. Oh, yeah. Oh,
Probably
yeah.
are
Yeah.
few.
There's no.
But what you're saying, though, I guess, is the positioning of the product is not. It's not a replacement for.
No.
It's a
Yeah, it's a supplement.
adding it into the mix.
Yeah.
To give it the microbes that makes a total sense.
Yeah.
And from a competition point of view, like, can you speak about other companies doing this, or are you leading?
Yeah, so I mean, it's difficult for us. We don't really. There are many infant probiotics out there, but we don't think of many of them as particular competitors often because they've not been clinically validated. They've not been particularly rationally designed.
Yep.
And when we culture them in house, we're getting different strains and what they say. So we're not at the moment really thinking of them as
really
our competitors. And also right now we're also
scary.
Yeah, it's scary.
That's scary.
But we're also trying to target preterm populations as well in hospitals. And so actually from that number, you massively have a lot less people interested. But the probiotics that are out there are normally 1 or 2 strains. Sometimes they're 3 bacteria strains, which I just don't think is sufficient for covering all the different babies and the different environments they're from and different ethnicities and geographies. And, I just don't think it doesn't make sense to me that just 1 or 2 strains can have this amazing effect. So that's really where we're planning to differentiate.
Because some people sit on the, there's a spectrum of, single strain all the way to what we're doing, which is pooled microbiomes derived from people. So it's like higher diversity than any donor.
Yes.
And some people say, well, we think the single strains are sufficient. And
I think that's a problem with a lot of these clinical trials already with preterm infants and NEC is that they only seem to work if these babies are lucky enough to be given breast milk. If they're not, and so the breast milk we know already has got the microbiome in them. So, a lot of the best ones of, so I think that these bacteria, you need to get the right environment as well.
You need to get environment. So you're in the consortia side.
We are, yes. I mean, yeah, essentially.
Rather than the single.
It makes sense. I mean, it's not only just for how we're trying to, all our different bacteria will have slightly different functions, but we do need a bit of redundancy, we think, for these strains to take because there is differences between infants. So you just need, maybe this bacterium will work for one infant, it won't work for the other one, but if you've got a similar one, it might work for that one as well. Trying to be a bit more broad.
So the redundancy piece is that they might not all be absolutely necessary, but it may be the case that some take up in some recipient microbiomes and don't get booted out by the immune system, whereas in others it's a different picture. And if you have enough, you're likely to get an effect in every single patient.
That's what we hope. Yeah.
Something like that.
Yeah.
That makes a lot of sense. Are you able to talk about at this stage what the vision is for the business, like where you want to take it? And
I mean, we really just want to, I think something that we find all the time when we talk to parents is that there's so much unknown about, people sort of know that, C-section, maybe I shouldn't have done it, but I can help it. And obviously I would never say to someone, don't get a C-section because I think how many babies would have just not survived if it wasn't a C-section. So that's mental to just be, discourage women from getting C-sections if they need to.
100%.
But they're very worried about, but what's the case of the microbiome? And or same as antibiotics. I feel like, should I have said no to the antibiotics?
No, you should have said It's like if they're clinically indicated, then please do.
Exactly. But there's so much doubt, and I think as awareness of microbiomes rate is getting greater. I think it's important for people to have a place to go to get useful information. So we really want to be positioned as a place where you can get clinically valid advice as well as products that can help for all parents. We think that, microbiome is so important in so many different diseases and, long-term great health outcomes too. And it starts as soon as you're born. So for us, we just really want to think, we think that, if we can get all of these babies having the right microbiome immediately, that's going to have a huge impact on model health and really improve the quality of, the next generation's health. And that's where we want to be. That's our vision. And our mission is really just to support parents and to achieve that.
And you took on some funding last year, is that right?
Yes.
Or the year before last? Last year?
Yes.
How was that process?
So half of our funding is from grants. So we've won some grants from Innovate UK and also from iUK. And that was hard. It took a while learning how to write grants and getting enough data to persuade the reviewers as well how to do it. We failed a lot in winning grants. I don't know if you've tried the grant grant writing route.
We have, I mean, we're doing less and less of it now because the amount of money we're spending per month is getting to the stage where it needs to be a huge grant for it to move the needle. But the most recent one was the grant that Imperial College submitted with us as a co-applicant to the Medical Research Council.
Yes.
That was like a £2 million pound plus grant award. And we've been knocked back many times from grant applications for sure.
Yeah.
Even with what we thought were really strong proposals.
Yeah.
Not saying we wouldn't do another one. I think we probably will. And it just has to be sufficiently sized for it to move the needle for us. Because as you know, it's a massive time commitment to produce a good grant.
Oh yeah. And it's you put your all in it, and you get really excited. And we've had a lot of failure of grants, but when you finally win one, that was a game changer, because it meant that we could demonstrate this to investors. You could go to investors and be like, look, we've won a grant. Their due diligence is basically done, because it has. Because I mean, your grant reviewers are experts from all walks of biotech and doctors and. So, I mean, it really has been done if you've successfully won one.
Yep.
And from that, we managed to get, some investment from a VC and also some angels, angel investment, which was really great. And it's really, they're really helpful advisors that we've got. And we've managed to build a board now of different people from.
Very important.
Yeah, making different products. So, it's been really exciting.
Good.
And we've got enough runway for a couple of years.
Good.
But,
lucky you. Not many companies can say that at the moment.
Well, as long as we keep fairly small. We actually raised at the perfect time. We were really, really lucky.
Good.
Because we closed it in April.
Very good.
And
how many employees are there?
Yes. Oh, we're pretty small. We're still trying to outsource a lot, to be honest. We're 6 at the moment. We sort of wax and wane a bit. Mm-hmm. But we're 6 at the moment.
That's good. And that explains why the costs are not like.
Well, we're also really lucky because we're collaborating with an academic at the Institute of Child Health, which is the research arm of Great Ormond Street Hospital.
Oh, perfect.
So we've got collaboration with her, and she's really great. She's giving us her lab space at a fairly reduced price.
Fantastic.
Because they're really supportive. UCL's a great university. That's where I did my PhD, and so very supportive of our venture. And we've got all these
alumni
in our hands. Yeah, exactly.
Alumni smashing it.
So that's been very helpful for us. Yeah.
Good. Very good. Well, I think we've gone over an hour now.
Oh.
And I don't know how long we've actually been going on for, but we did say we would try and keep quite tight for time, and I was given strict instructions by you not to talk.
No,
I'm only joking. I'm just
Jay, you need to go to your training. I know your agenda.
I'm just winding you up. I'm totally
very wound up now.
No, you've been a great guest, and there's lots more things to talk about, so maybe we can go on again. And you can interview me if you want and ask me lots of hard questions.
Grill you.
But this is great. I wish you all the best with BoobyBiome. If people want to follow your company's progress, where do they go?
Oh, yes, we're on Instagram at BoobyBiome, as well as, of course, LinkedIn. We're not sure about Twitter, but we do have a Twitter account. We don't use it much.
TikTok? No.
TikTok is where, we're going to be big on TikTok. We've got great big ambitions, but we're still early on TikTok. So most of the content is really our Instagram page. Yeah.
Well, thank you so much. Pleasure.
Thank you so much. It was great.
Thank you. Good.